Hello. So this is my master's thesis defense, third reiteration of my talk. I have particularly made changes to the second chapter, my kind of explanation. So pay particular attention to that, mainly Stephen. So here is my talk. Let me start the screen share. And for anyone that listened, thank you for doing that. Whoa, hold on. Wrong desktop. Okay. Okay, so today I'm going to talk about my master's thesis research, which is examining DNA methylation variation in gametes and larvae of the Pacific oyster, Chrysostrea gigas. So today I'm going to start by giving some background information on epigenetics, specifically focusing on DNA methylation. Then I'll present some information on what we already know about DNA methylation patterns and functions in various species. Then I'll talk about the results of my thesis, which has two different parts. The first part characterizes DNA methylation on a genome-wide scale in oysters in order to understand the functional role of methylation. And this included examining the relationship between DNA methylation and gene expression in the species. And the second part characterized DNA methylation patterns during oyster development, which included examining the degree of DNA methylation, heritability, and variability of these marks. So the fixed epi means at or upon. Therefore, epigenetics describes DNA modifications that change gene expression without altering the underlying nucleotide sequence. And this involves mechanisms other than mutations to the DNA sequence. And epigenetic changes can switch genes on or off. Examples of such epigenetic mechanisms are histone modifications, and histones are the proteins that package DNA into nucleosomes for compact cellular organization. And these proteins can be modified by acetylation, methylation, and phosphorylation. And DNA methylation is another epigenetic mechanism, and both of these mechanisms serve to regulate gene expression without altering the underlying nucleotides or DNA sequence. DNA methylation is the most well understood epigenetic mechanism, and it involves the addition of a methyl group to DNA. It's highly specific and will typically happen in a region in which a cytosine nucleotide is located next to a guanine linked by a phosphate, and this is therefore called a CpG site or dinucleotide. An insertion of this methyl group will change both the appearance and the structure of DNA, which will modify a gene's interaction with a cell's nucleus, which is needed for transcription. And interestingly, DNA methylation in organisms is diverse, variable among species, and can even change genome functions under external influences. DNA methylation has many important regulatory functions in gene expression in a variety of different organisms. To name just a few, it's involved in embryonic development. It's an important regulator of developmental processes. It's involved in genomic imprinting, which is a phenomenon in which genes are expressed in a parent of origin specific manner. In sex ratios, so in European sea bass, methylation of the gonadal aromatase promoter is involved in temperature-dependent sex determination, as well as diseases. So aberrant DNA methylation is a feature of a number of human diseases, including cancer. And there's also a wide range of DNA methylation patterns. So vertebrate species like humans will exhibit what's called global methylation, in which their genomes are between 70 and 80% methylated. And this figure is depicting a vertebrate genome that would exhibit that global DNA methylation landscape. The gray regions will indicate methylated domains, and yellow indicates unmethylated domains. So only small regions of the genome are unmethylated or transcriptionally active. However, invertebrate species display limited or no methylation in their genomes. Some of our common model invertebrates lack methylation completely, such as Drosophila and C. elegans. And we found that invertebrates such as oysters contain what's called a mosaic pattern. And this figure on the bottom is depicting what a typical mosaic DNA methylation pattern would look like. It's characteristic of most invertebrates in which you have heavily methylated or the gray regions that are dispersed within domains that are methylation-free, the yellow regions. So these differences in methylation patterns illustrate the diversity of patterns, but also highlights our gaps in understanding the evolutionary and functional significance of methylation. And not only are DNA methylation amounts different across taxa, but the function of methylation differs as well. So in vertebrates, DNA methylation is one mechanism for suppressing or silencing gene transcription and will thereby prevent gene expression. So methylation is found throughout the genome in both genes and promoter regions, which are regions of the DNA that initiate transcription of a particular gene. And methylation among promoter regions will block the areas that transcription factors would bind. However, like I previously said, most of what we know about methylation is from mammals and plants, and less is known about invertebrate species. But most invertebrate genomes contain methylation that's located within genes, but the function still remains unclear. So most of what we know about methylation is from mammals, less is known about invertebrates, and we see very dramatic effects of methylation on mammals. And in mice, it's been shown that epigenetics can affect gene expression. And a great visual example of this can be seen in the agouti mice. So these are two female mice that are genetically identical, but they have different DNA methylation patterns in their agouti gene, which will affect gene expression and therefore give rise to these two very different phenotypes. So specifically in the small brown phenotype, the agouti genes are methylated and therefore turned off, which will prevent transcription. However, in the yellow obese mice, the agouti genes are not methylated, and these genes are expressed or turned on. And when the agouti genes are expressed, the mouse is more prone to diseases like cancer and diabetes. And research in the Waterland and Jurtle lab has found that the activation of a single gene due to a lack of methylation can cause dramatic changes in phenotype like seen here. And they therefore confirmed that DNA methylation plays a role in regulating gene expression. DNA methylation has also shown to be heritable and can be maintained and passed on to offspring. This was demonstrated under natural conditions in the plant toadflax or Linaria vulgaris. So specifically, wild-type and peloric toadflax plants are genetically identical, but they have different shapes of their flowers, which is thought to be beneficial for different pollination strategies. And these two flower variants have different methylation patterns in a single gene, which is heavily methylated or inactive in the peloric variant. And peloric toadflax can pass on this heavy methylation status to its offspring. So an abnormal flower structure will continue on to the next generation. And this is an example of methylation patterns being inherited. But this type of transgenerational epigenetics has been well documented in plants, but it's more difficult to demonstrate in other organisms. So the background I just provided summarizes what we already know about DNA methylation. And I mentioned that a majority of this information comes from studies done on mammals and plants, and much less is known about methylation in invertebrate species like oysters. But some previous work in our lab has examined DNA methylation in oysters. This is research done by Mackenzie Gavery, who's a former grad student in our lab. And to summarize what she found about methylation in oysters, she used an in silico and experimental analysis to show that methylation is in fact present in genes, and that methylation levels can change with gene functions. However, this work was only predicting methylation status from the oyster transcriptome, or the set of all the RNA molecules. And with the release of the oyster genome in 2012, this greatly facilitated my research, as I was able to quantitatively examine DNA methylation at a genome-wide scale. So my research aimed at characterizing and understanding this epigenetic mark in oysters, and this was important as limited studies have focused on invertebrates. And currently, there isn't a complete understanding of how DNA methylation regulates genome activity in invertebrates, although it does appear to be different from the mechanisms we observe in mammals and is likely variable across species. So my approach was split into two different parts. The first part was characterizing genome-wide DNA methylation profiles in male gamete cells using whole genome bisulfite sequencing. And we were particularly interested in the relationship between methylation and gene expression. The second part was examining DNA methylation during oyster development. So while we know that methylation has important regulatory functions in mammalian development, we still don't understand the roles of methylation during invertebrate development. And a major component of this is understanding if DNA methylation is inherited between organisms and to what extent DNA methylation patterns vary between individuals. So the approach I used for my research is to use what's considered the gold standard for examining DNA methylation. This is called whole genome bisulfite sequencing. And this involves genomic sequencing of bisulfite-treated DNA. So for this method, DNA is treated with sodium bisulfite, which will convert cytosine residues to uracil, but leave methylated cytosine nucleotides unaffected. Therefore, this bisulfite treatment is introducing specific changes in the DNA sequence that depends on the methylation status of individual cytosines. And after sequencing, I'll map my results back to the oyster genome, and I can conclude that any cytosines that are remaining are in fact methylated. In using this approach, I can characterize methylation on a genome-wide scale while still obtaining coverage at a single base pair resolution. So my analysis is reduced to distinguishing cytosines that's resulting from bisulfite conversion. But we're able to differentiate these marks from single nucleotide polymorphisms by using information from the complementary DNA strand. So the first part of my research was completely novel, as it was the first time genome-wide methylation was examined in oysters, which allowed us to see how methylation was distributed in the genome, and also look closely at the relationships between DNA methylation and gene expression patterns. So for this research, I performed high-throughput whole genome bisulfite sequencing on oyster male gamete tissue to provide a comprehensive view and reveal methylation patterns. And to understand potential functions of DNA methylation, I examined the relationship between methylation and gene expression. And to examine this relationship, I used an approach called RNA-seq, or whole transcriptome shotgun sequencing. And basically, this involves taking a snapshot of the RNA presence and quantity from the oyster genome at a given moment in time, so we can look at the relationships between epigenetic and genetic variations. So using the oyster reference genome for mapping, I obtained information for about 7.64 million CpGs. This is showing a histogram looking at the distribution of methylation, and we see a bimodal distribution, and that a majority of the oyster genome is unmethylated. But we see that the genome does in fact contain some methylation, so we wanted to describe where this was occurring. So to summarize where exactly methylated cytosines are distributed in the oyster genome, these are pie charts showing the genome-wide distribution of methylation within genomic regions. And the figure is showing the proportion of methylated or unmethylated regions as percentage contributions to specific oyster genomic regions. So we classified CG loci as being either heavily or sparsely methylated, defined as having a methylation ratio greater than 0, or unmethylated regions, which had a ratio equal to 0. And the genomic regions are also scaled according to their contribution to the entire oyster genome. So a larger pie is representative of a larger region in the oyster genome. And the takeaway message from this figure is that methylation is occurring primarily within genes, in exons and introns. You can see that the dark blue slices are comparatively larger than that among other regions of the oyster genome. So exons and introns were about 35 and 28% methylated, compared with other genomic regions, which were only about 15% methylated. And we were able to determine that overall 15% of the CpG dinucleotides are methylated in the oyster genome, and this number is dramatically lower than what we see among vertebrate species, in which their genomes are heavily methylated between 70 and 80%, but it is comparable with other invertebrate species. And from the pies I showed you on the previous slide, we found that a majority of methylated regions are occurring among genes, or exons and introns. We were also interested in using this data to identify any relationships between methylation and gene expression. An analysis of our methylation and RNA-seq data revealed a positive association between methylation status within promoter regions and expression. This figure is showing the proportion of methylation on a per-gene basis for promoter regions, or regions of DNA that it will initiate transcription of a particular gene. And I've divided our RNA-seq data into high and low expression categories based on expression values. And then I calculated an average percent methylation for promoters within these categories. And we see a positive correlation between DNA methylation and promoter expression. And this was interesting because previous research has indicated the opposite, in which a negative correlation exists between DNA methylation and expression levels. So if we go back to our introductory diagrams, previous research in mammals has shown that the primary function of DNA methylation is to suppress gene expression through increased promoter DNA methylation. So methylation found in promoter regions would block the areas that transcription factors would bind, therefore being associated with a decrease in gene expression or transcriptional silencing. However, this doesn't seem to be the case in oysters, as we found that methylation in promoter regions is being associated with transcriptional activity or gene expression. And based on our findings that methylation is primarily located among genes, and if methylation is in fact regulating the oyster genome, we'd also expect to see a relationship between methylation and gene expression. And we do see a positive correlation between DNA methylation and gene expression, in which genes with high expression levels had significantly more methylation, indicating that DNA methylation may be involved in gene regulatory activity. So using the oyster as an invertebrate model, we saw that methylation was primarily located among genes and is associated with transcriptional activity or gene expression. And to explore some potential reasons for this, it's possible that those genes with high methylation are housekeeping genes that are involved in basic metabolic functions. And these genes are highly expressed in the oyster genome in order to allow for efficient expression or regulation of these genes that are necessary for survival. Therefore, these housekeeping genes involved in maintaining essential cellular functions would need to be efficiently expressed. And those genes that are less methylated will be those involved in environmental response pathways. These might be genes that change in response to an environmental stressor and would therefore not need to be consistently expressed in the oyster genome. And this would help reduce any unwanted or unnecessary transcriptional noise in the oyster genome so that genes required for maintaining basic cellular functions can exhibit a consistent expression. Together, this would indicate that methylation may not necessarily be controlling gene expression directly, but is rather controlling the amount of transcriptional noise that's present in the oyster genome. So this study was of interest because most previous research has concerned mammals, and we provided a comprehensive characterization of methylation in an invertebrate. This was the first high-resolution methylation map that's been generated for a molluscan species, which allowed us to examine both the distribution and potential regulatory functions of DNA methylation in the oyster genome. To summarize data from this study, I found that overall 15% of CpGs were methylated in the oyster male gamete genome. Again, this amount is much lower than what we see among vertebrate species with heavy genome methylation, but it is comparable with other invertebrate species. We were also able to provide a comprehensive profile of where DNA methylation occurs across the oyster genome, with it being primarily located among genes or exons and introns. And finally, based on our gene expression data, we saw a positive relationship between methylation status and gene expression in both promoter regions and genes. And I believe that those core housekeeping genes involved in maintaining basic cellular functions would need to be heavily methylated and constantly expressed in the oyster genome, whereas a lack of methylation in genes involved in responding to environmental conditions would allow transcriptional variation. So this indicates that methylation may not necessarily be controlling gene expression directly, but is rather controlling the amount of transcriptional noise that's present in the oyster genome. So this research provided a comprehensive view of DNA methylation patterning in oysters, as well as evidence for a relationship between methylation and gene expression. However, there were still many things we didn't know about methylation in oysters, including if these patterns are heritable. So the objectives of our next project were to examine to what degree DNA methylation patterns are heritable, variable between individuals, and changing during oyster development. So to examine the degree of DNA methylation heritability in oysters, this is a schematic showing our experimental design. We fertilized the eggs from a single female with the sperm from 2 males. And it's worth noting here that the 2 males were fraternal. They were full siblings, meaning that genetic variation was likely minimized. And we also used a single female in the experiment to reduce any potential variability. The fertilized eggs were placed in 2 separate tanks according to the male oyster. And we took samples of oyster larvae at 72 and 120 hours post-fertilization, and then performed whole genome bisulfite sequencing to examine methylation patterns from these 3 developmental stages. So we're looking at sperm and larval oysters at 72 and 120 hours post-fertilization from these 2 oyster lineages. So to begin to investigate the role of DNA methylation in oysters during development, we were first interested in exploring parental transmission of methylation patterns and seeing if these patterns were heritable between parents and their offspring. And this involved comparing methylation patterns between the male sperm samples and their larvae at 72 and 120 hours post-fertilization. Therefore, we're examining differences in methylation among these 2 oyster lineages. And one way to begin looking at the distribution of methylation patterns is to use genomic visualization tools. This is a program called Integrative Genomics Viewer. So we can begin to visualize patterns of methylated and unmethylated regions in the oyster genome. This is showing a single scaffold of the genome, or about a 200 kb region, containing 6 different tracks for each of the oyster sperm or larvae samples I examined. And the numeric prefix here refers to the oyster lineage in which the sample originated from. So each bar here represents percent methylation for an individual CG loci. And we see that overall, it looks like there's about 15% genome methylation, which correlates with what we previously found in the oyster male gamete from the first part of my research. And we can also begin to visualize patterns of differential methylation between these 2 oyster lineages. And one of the main findings from this study are that oyster lineages were actually more similar in their methylation patterns rather than developmental stages. This is a dendrogram of the male sperm and larvae using Pearson's correlation distance. So samples that are grouped together are more similar in their genome-wide methylation profiles. So this finding was interesting because we expected the sperm to be similar, as it's a single cell type and larvae are mixed tissues. And in other organisms, DNA methylation has shown to be variable across different tissue types. And like I also said, the sperm samples were full siblings, so we would assume a lack of genetic variation between these 2 individuals. So this was the first indication that there appear to be different epigenetic patterns that may differ among oyster lineages. And in fact, the two male sperm samples actually had the least similarity in their methylation patterns out of all of our pairwise comparisons. So specifically, I found that about 3,000 common loci between the sperm samples contained a methylation difference of at least 90%. So to examine where in the oyster genome these differences were located, I identified loci that significantly differed in methylation levels between these two oyster lineages. And these loci are termed differentially methylated loci, or DMLs. And there were 188 total loci that differed in methylation levels across these two lineages. And these differentially methylated loci were classified with respect to oyster genomic regions like exon, intron, promoter regions, and transposable elements. And while we're only examining regions of the oyster genome that have been annotated, we do see that a majority of these DMLs are present among genes, or exons and introns, and also transposable elements. However, when examining the distribution of these DMLs, we need to consider how many CGs are located within each of these genomic regions. So we also need to take into account the distribution of all the CGs in the oyster genome as a comparison. So if the distribution of these differentially methylated loci were random, we would expect a similar pattern as seen in the column showing the distribution of all CGs in the oyster genome. And interestingly, we found a high proportion of differentially methylated loci among transposable elements when compared to the distribution of all the CG dinucleotides in the oyster genome. And this difference was statistically significant. We also see that the distribution of differentially methylated loci among genomic regions like exons or introns is not significantly different than the distribution of the CGs in the oyster genome. So this indicates that methylation may be enhancing randomness by targeting transposable elements rather than genes. So what I'm proposing is that differences in methylation we're observing may be random, but a change in methylation or gene expression among functional genes that are core to oyster survival would result in a lethal situation. So it would therefore be beneficial for genes, or promoter regions, exons, and introns, to have consistent methylation patterns rather than being differentially methylated. However, a change in methylation among transposable elements could potentially be a lethal situation if a transposable element insertion occurs in a core functional gene and disrupts gene expression. But transposable element mobility could also generate new genetic variation, which would be important for organisms like oysters, in which populations are living in stressful intertidal areas and might experience abrupt changes in environmental conditions. Therefore, I believe that these differential methylation patterns are occurring among transposable elements because differences in methylation can be random. So it isn't crucial if transposable elements are either methylated or unmethylated. However, it would be crucial for the methylation status of functional genes to be consistent. So what exactly are transposable elements? They are DNA sequences that have the ability to change their position in the genome. So they're also known as jumping genes because they can jump to different locations within a genome. And transposable elements are commonly known to be silenced by epigenetic mechanisms like DNA methylation. So methylation will silence both the expression and mobility of transposable elements. For this experiment, I was also interested in exploring any developmental patterns of DNA methylation. And this involved comparing methylation patterns between developmental stages. So this gives us information about the methylation changes that are occurring during oyster development by looking at these 3 developmental stages: the sperm and 2 larval oysters at 72 and 120 hours post-fertilization. And by examining overall DNA methylation levels during the first week of oyster development, these are some box plots showing the average methylation levels for each of the 3 developmental stages I examined. And we see that DNA methylation levels do not vary significantly during developmental stages. But to examine any methylation differences that may be occurring during oyster development, we again classified differentially methylated loci, or DMLs, and I found that there were 157 loci that differed in methylation levels at these different developmental stages. So for example, comparing the sperm to the larvae at 72 hours post-fertilization. And these differentially methylated loci were classified with respect to oyster genomic regions like exon, introns, promoter regions, and transposable elements. And we see that a majority of these differentially methylated loci are present among introns and promoter regions. Again, we do need to take into account the distribution of all the CGs in the oyster genome as a comparison to consider how many CGs are located in each of these genomic regions. In examining this distribution, we found a significant difference in differentially methylated loci during oyster development located in promoter regions relative to the distribution of the CPGs in the oyster genome. So this phenomenon isn't what we observed from the first part of my research, examining relationships between methylation and gene expression. So if you remember from our previous diagrams, we found that consistently high methylation patterns among promoter regions may not necessarily be controlling gene expression directly, but rather controlling the amount of transcriptional noise present in the oyster genome. However, we are seeing differential methylation among promoter regions during oyster developmental stages, which suggests that promoter methylation may play a role in oyster development. And this finding is consistent with previous research on oysters by Riviere, who found that DNA methylation may specifically control expression of some oyster developmental genes through methylation of these promoter regions. So some conclusions from this second chapter. Our research suggests that DNA methylation patterns are inherited, as methylation patterns were most similar between males and their offspring. I was surprised to find the presence of lineage-specific methylation patterns. This is due to the fact that I expected the two sperm samples to be the most similar in their methylation patterns, as they were full siblings and male gamete single cell type. Found a high proportion of differentially methylated loci among lineages present among transposable elements compared to the distribution of all the CGs in the oyster genome. And I believe that these differences in methylation may be enhancing randomness by targeting transposable elements rather than genes. We also didn't observe any dramatic differences in methylation during oyster development or between individuals. I found that DNA methylation levels were similar across all of my samples, indicating that methylation does not significantly vary between cell types and life history stages in oysters. However, when examining the differences that do exist between these developmental stages, I found differential methylation among promoter regions. So this suggests that promoter methylation may play a role in oyster development. Overall, my research found a relationship between methylation status and gene expression, and I'm suggesting that DNA methylation may not directly be regulating gene activity, but rather controlling the amount of transcriptional noise that's present in the oyster genome. I've also shown that methylation patterns are likely inherited, as they were different between oyster lineages. And we also found differences in methylation among lineages to be present among transposable elements. And I propose that this methylation may be enhancing randomness by targeting transposable elements rather than genes. So this research could have significant implications for the way organisms may respond to changing environments, particularly with respect to climate change. Oysters are common models for studying adaptation of aquatic species to climate change, and epigenetic mechanisms like DNA methylation may explain how oysters are able to handle surviving environmental change through a mechanism that can't be explained by genetics alone. DNA methylation could also underlie processes that allow species adaptation over evolutionary time. So if methylation is repeated across generations, successful methylation patterns could induce permanent changes in the genome. And therefore, this research could completely change the way we think about certain evolutionary concepts. And that is it. Thanks for listening.